Alright, so I know that the beginning there was a little bit silly, but the reason why I did that was that I wanted to get a point across. Just like in my little introduction, I made my dog from a recipe. All living things have a recipe of their own, and that recipe, or that set of instructions, is called DNA. In this unit, we're going to look at how DNA is used to make protein, and we're going to go over DNA again because it is so important, and even though you already have some background about it, I'm going to add on even more. If you remember, so you don't have to write yet, DNA is a nucleic acid, one of the 4 main biomolecules of life. It's called deoxyribonucleic acid, and we find it in the nucleus. It's a set of instructions that codes for protein. And something that is so important to understand, the big idea for this unit, and maybe you want to write this down at the top of LO1, is that this whole unit from LO1 to LO6 or LO7 or how many ever I have in this unit is for how we get from DNA to protein. It's the basis of how an organism gets made. The shape of DNA was discovered in the 1950s, so actually not too long ago. One of the key components was provided by Rosalind Franklin. She was a female scientist, which at that time was very rare, who used X-ray crystallography to take pictures of DNA. You can see her most famous photograph at the bottom on the right. She was very, very close to discovering what the shape of DNA actually was, and her photograph was passed on, without her permission, to James Watson, and his partner was Francis Crick. And with this photograph, James Watson and Francis Crick understood how DNA was set up and quickly got the information published, and so they essentially won the race for figuring out how DNA was shaped. While a lot of the work came from Rosalind Franklin, she was not really given the credit back then. There's a whole controversy surrounding her, and if you're interested, you can just search her name and find she has a biography. There's lots of information online about how she should have deserved more credit and why, and she didn't. But anyway, Watson and Crick are given the credit for discovering the shape of DNA. If you remember the basics here, DNA is a code for amino acids. Amino acids are what make up protein. They're like beads on a string. The beads would be the amino acids. The whole necklace would be a protein. Proteins are needed for just about everything in your body. It's more than just what's in meat. You need proteins to send signals to cells. You need proteins to move things around your body. They are so widespread and so important that if you didn't have DNA, you literally would not be alive. DNA is located, once again, as I mentioned before, in the nucleus of the cell. The monomer of DNA is called a nucleotide. You put lots of nucleotides together, you get a nucleic acid. Remember, with proteins, the monomer is an amino acid. You put them together, you get a protein. Nucleotides are the P, S, and B things that we talked about before. P is phosphate, we have a sugar, and 4 options for a nitrogenous base. You can have A, T, G, or C. Adenine, thymine, cytosine, and guanine. DNA has 4 properties that we already mentioned before. It's double-stranded, anti-parallel, meaning the 2 strands run in opposite directions. It's a helix, it's a wound ladder shape, and it's complementary. The 2 strands pair together in a very specific way. A goes with T, G goes with C. Something new, a gene, spelled differently than the genes that you wear, G-E-N-E. A gene is a very small section of DNA that we're going to read, copy, and use that copy to understand the code and make a protein out of it. So in this very simple diagram here, this strand of DNA has 1 gene, 2 genes in this very small section. So there are lots of genes per chromosome, per strand of DNA. Let's practice one more time, just like we did in the last unit, making the other strand of DNA. We're going to have to do something similar with RNA in LO3 and LO4, but for now, let's work with this other strand of DNA. Remember to label your ends, because the ends run opposite directions, and then A pairs with T, G pairs with C. See if you can figure it out, and then come back and check your answer. You should have AGA, CGG, TCT, TCA, AGC, CC. And then also have labeled the ends in the opposite direction, 3' and 5'. That is the end of LO1. Next, you're going to complete an online activity that looks at how DNA fingerprinting, because everybody's DNA is unique, can be used to solve crimes. And you could also use it to do paternity testing and other things. And then after that, you're going to be able to extract DNA, or take it out of strawberries, and see what a glob of DNA actually looks like. There is also an extra credit opportunity available in the crate. And if you have any questions about what's happened in this video, make sure that you come see me.